In this video, we're going to study the components of an experiment in more depth. We know an experiment is the only type of primary data collection method where you can definitely try to show cause and effect relationships. We do this by manipulating attributes of the study participants and trying to observe what the consequences are. In order to set up an effective experiment, there are 5 components you have to consider. Number 1. Factors are the attributes that are manipulated in the experiment. The manipulation can occur in various ways, and the various values that you do manipulate to are called the levels of the factor. These levels are assigned to certain individuals in the study. Those individuals or items being studied are called experimental units when they are objects, or subjects when they are people. The specific combination of factor levels that get assigned to a specific subject or experimental unit is called the person's treatment. You can think about this just like in the medical setting. And finally, the variable measured is called the response variable. The response variable is just another name for the dependent variable, whereas the factors being manipulated is another name for the independent variable. So we're still focused on establishing independent and dependent variables. We're just going to do it with a little bit more detail than we did in the last couple examples. Next, we're going to look at each of these components for a specific example. We want to test the effects of lighting on workers' production levels among sewing machine operators. We're going to split the group of 60 operators into 3 groups of 20 at random. One group of 20 will work with 60-watt lights. Another group of 20 will have the intensity change to 75 watts, and the last group will go to 100 watts. After the different groups have worked with these varying light conditions for 15 days, we will look at the total production to see if there's a difference between the pre-production levels, which were recorded every day, and the post-production levels. So first you need to ask yourself, what is the factor? To answer this question, you need to look at what is the independent variable that is being changed. So what are we changing for these various people in order to see if there's an impact on their production? The answer to this is the lighting. The factor being manipulated here is the lighting levels. For the exact same example, we can ask ourselves, what are the different levels of lighting? We know the factor being manipulated is the lighting, but to what levels? In this case, we have 3 groups, so the levels are the lighting for the 3 groups. 60 watts, 75 watts, and 100 watts are the 3 different levels for this specific experiment. Next, what is or are the experimental units? In this case, you need to look at who or what the lighting is being manipulated for. In this case, we're talking about the production of people operating sewing machines and if the lighting affects them. When studying people, we call the experimental units the subjects. So the answer to this one is that the subjects are sewing machine operators. We are influencing the lighting for sewing machine operators. Number 4, what are the treatments? Treatments are the specific levels that each group gets assigned. So we have Group 1 getting 60 watts. The treatment for Group 2, we're told they get 75 watts of lighting. And the treatment for Group 3 is 100 watts of lighting. These are our 3 treatment groups. And finally, what are we trying to measure based off of the different lighting levels? We're trying to see what it has an impact on. In this case, it is the production. We're trying to see if production measured every day is impacted by lighting. So essentially, we're trying to see if lighting, the independent variable that changes, impacts your production. Production is our response, which is just another name for the dependent variable in this experiment. Make sure you try identifying these components for the learning check example on cookies. Next, we're also going to talk about the 4 principles of experimental design. Once you have established the components, there's also some principles you must follow to make sure your experiment is designed correctly. The first is to randomize. Second is to control. Third is to replicate or repeat observations. And fourth is you can have what's called a block, which means group similar subjects together. We're going to look at each of these in detail. First, randomization. That just means you assign treatments to people randomly. Randomization allows us to equalize the effects of unknown or uncontrollable sources, like if someone's feeling really tired that day. We randomize people to groups, so hopefully like all the tired people aren't all in the same group, for example. Randomization makes it possible to use inference methods, basically to know the results of the experiment will be more valid and aren't related to impacts other than the lighting, which we really are trying to see if lighting impacts production, not all these other things that could be happening in people's lives. So randomization gives us protection from those things that you can't predict or know what's happening in everyone's life. The saying goes, control what you can, randomize the rest. So we control the lighting, which we can control. We try to randomize other effects by assigning people randomly to different lighting groups, for example. Number 2, control. You'd want to make the factors you're not testing as similar as possible. So the only difference between the group should be what you're manipulating. So for our previous example, the only difference between the group should be the lighting. Other things like humidity or the working hours in the shift should be the same for all participants. So the only thing that we're trying to change for them is the lighting, so we can see if the differences in production really come from lighting. There's also a second meaning of control. A controlled treatment is a special treatment class to mark the baseline of the study. This means that there's no change. So for the lighting example, before the researchers came in, all the workers would be working at 60 watts. Notice that one group stayed working at 60 watts with no change. They would be called the control group because they're still working in the same conditions. Another factor related to control is blinding and using placebos. So blinding is deliberate withholding of the treatment details from individuals. So this can help you avoid bias. So if people know that they are getting certain treatment, it could influence how they feel. So we can blind the people who are actually getting the different levels of the factor, like the subject. So in this case, you might not tell the sewing machine operators who's working under what lighting conditions. That would be blinding. You can also blind the people who evaluate the results. This usually happens in medical experiments where both the patient and the doctor would not know which treatment the patient is getting. Only the researcher or the experimenter would know. This is because if the doctor knows what treatment you're having, they could also influence how you feel, either on purpose or not on purpose. So by not letting either the doctor or the patient know, it can help to avoid bias. Something related is including a placebo. A placebo is a fake treatment that looks just like the real treatment being tested. Some more information on blinding, as I mentioned, is if only the patient doesn't know what medication they're receiving, that's called a single blind. But if both the patient and the doctor, for example, don't know like who's getting the real pills and who's getting the placebo or the fake pills that we can use to compare the results of the real pills to, that's called a double blind. Principle number 3 is to replicate. You shouldn't just do the experiment once for each group. So for our lighting example, we should not have one person working under each lighting condition. You need more than one observation at each level. For the lighting example, we have 20 people in each group. So that's known as replication. We have more than one person in each group. If the number of replicants is the same for each treatment group, then it's called balanced. For lighting, we have 20 in each group. That's the exact same number in every group, so it is balanced. And finally, block. Block means grouping similar subjects together based on a factor that you cannot control. Examples of this are someone's gender, their ethnicity, or marital status, for example. For our lighting experiment, if you believe that gender, for example, could also have an impact on production, you can't randomly assign people to conditions like being male and female. They automatically come to you with a gender. So what you would do is break them into 2 groups. If you think gender has an impact, you could measure the production for all of females separate for all the males, or separate for all the people who identify as non-binary, for example, and then look at the impacts for those separate groups. A block essentially means running different experiments for the different groups. So if you split according to just male and female, that would essentially be like running two different examples for the two different groups. Please make sure you try the learning check example on cookies to again help you to establish the differences in wording for the question between these four principles of experimental design. Finally, we're going to examine two different types of experiments: lab experiments and field experiments. Lab experiments happen in an artificial setting where you can carefully control and manipulate the variables in order to establish very strong cause and effect relationships. Lab settings are often preferred because the experimenter can control other variables that might contaminate the variables under interest. So for example, if you were working in a lab, you can control other factors like humidity or working hours to make sure that the impact on your productivity is just related to the lighting more carefully than you could if people were in their original workplace. So in a lab, you can control these a bit easier. That means that lab experiments have high internal validity, which means you can be sure one variable causes an effect in the other because you carefully controlled all other things in the artificial setting. However, the external validity is low. This means you can't be sure if the results you found in the lab will also be true in the real work setting because you aren't working in the real work setting. Another example is if you want to examine if people who are paid piece rate instead of hourly rates are more productive, you could do a lab experiment where different groups of people are paid in different ways and are asked to do a very simple task, like screw bolts into a plastic frame. If you find people who are paid piece rate produce more, you can establish that the method of payment does influence their productivity. However, you can't be sure if this would carry over to more complex settings in a real business because you only tested on a simple task, like screwing in bolts. In a field experiment, you conduct the experiment in the natural environment where events normally occur. The experimenter's ability to randomize people to groups or block could be limited if you're in a real work setting, but you can still try to manipulate conditions of work and set up a control group to compare to the others. Field experiments are actually the opposite of lab experiments because internal validity is low and external validity is high. This is because if you can find a relationship between variables in the organizational setting, you can easily generalize it to other organizational settings. You know it's true in the workplace, which means external validity is high. However, because you can't carefully control variables in a field environment like you can in a lab, it can be hard to prove the cause and effect relationship is just between the things under study. It could be true due to things that you could not control in the work setting. So this means the internal validity is low. In this video, we recapped the components of experiments, the principles of experiments, and types of experiments. Please continue with the lesson to learn about other factors that impact experiments.